Welcome to the video all about the plasma membrane. So even though this part of the video series is focusing on prokaryotic cells, it is important to note that all cells, whether they are prokaryotic or eukaryotic, have a plasma membrane. So the plasma membrane is the border of a cell. So think about a house. Houses look really different, right, on the outside and inside, kind of like cells. But all houses have walls, right? And it's those outer walls that say this is the house. So that's what the plasma membrane does. It is the border of the cell. And with that job or that title also comes the additional role of regulating what enters and exits the cell. So what that means is the plasma membrane is able to determine what molecules can go in the cell, what molecules can leave the cell, when things can enter and exit the cell, how many of each molecule can enter and exit the cell. So there's lots of regulation going on there. And it's able to do this because of its structure. Remember, structure and function always go hand in hand. So a phospholipid bilayer is the core component of the plasma membrane. So before we go into the details of this image, let's take a look at what a phospholipid looks like. Okay, for my computer, there we go. So a phospholipid has a hydrophilic head group and two hydrophobic tails. So there's a couple of science words there that you need to know. So hydrophilic means water loving. Hydro means water, philic means loving. So these head groups love water. They are very compatible with water. The two tails, though, they're fatty acid tails, are hydrophobic. That means they are water fearing. Okay, so again, hydro means water, phobic, like phobia, means fear. So these fatty acid tails do not like water. They are not compatible with watery environments. And that's really important because if you'll take a look at the picture below, notice how the two layers of phospholipids are arranged. Do you see how the fatty acid tails of this top layer of phospholipids are touching the tails of the bottom layer of fatty acid, the bottom layer of phospholipids? That's because the hydrophobic tails always want to be in touch with each other because they're compatible. Hydrophobic things like other hydrophobic things. So having the phospholipid bilayer be arranged like this is critical for its ability to regulate what goes in and out of the cell. And that's because it forms a hydrophobic barrier. Remember that most molecules are hydrophilic. That's why water can dissolve most things, because most things love water. So a lot of molecules are hydrophilic. That means they are not compatible with this hydrophobic barrier. That means they cannot easily get across the plasma membrane because they're not compatible with this core hydrophobic region. So how do molecules actually get in and out of the cell? That is going to be courtesy of transport proteins. And so if you look at this phospholipid bilayer, you see lots of things associated with it. We have some proteins like this blue one that are hanging out outside of the plasma membrane, right? It's only associated with this top layer of phospholipids. This purple one right here is also only associated with the top row of phospholipids, but it also has a carb attached to it. Some proteins are only associated with the bottom row of phospholipids. Other proteins, like this one here, are able to span the entire plasma membrane. We call these transmembrane proteins. But notice that this one is solid, but this transmembrane protein on the end here is hollow in the middle. So this one is a transport protein. Because it's hollow in the middle, that means molecules can use it as like a tunnel to get across the plasma membrane. So because you see some proteins and carbohydrates only associated with one layer of phospholipids, but not the other, we say that the phospholipid bilayer is asymmetrical. That means just the two layers are not completely identical to one another. It's also a flexible structure. We say it's fluid. That means these molecules can kind of move side to side a little bit. It's got to be flexible, right? Because cells are going to kind of bump into one another sometimes, and we don't want them to be so rigid that they just crack upon contact. But we don't want them so fluid, right, that things can just cross the membrane all by themselves all the time, right? We need that regulation. So it's important that the phospholipid bilayer, this plasma membrane, be fluid, but not too fluid, okay? So the proteins have different purposes. We're really focusing on these transport proteins here, but I do want to point out that the carbs and some of these other proteins that you only see on one side, mainly this top side, these are called peripheral proteins because they're on the periphery of the plasma membrane. These tend to be involved in cell-to-cell recognition. So just like we can recognize each other based on our physical attributes, cells can recognize other cells based on what proteins and carbohydrates are on the surface. Now, some of these peripheral proteins that are on the inside of the cell, these have more to do with like cell communication in terms of when the outside of the cell, maybe it detects something stressful in the environment. It can relay that information through like these transmembrane proteins right here, the solid ones. It can relay that information down. It then tells this peripheral protein, which will induce the cell to make some sort of change to try and survive whatever that stressor may be. Okay, so cells really are pretty complex, and the plasma membrane is key to its survival. Later, we're going to talk about certain antibiotics and antimicrobial drugs that target the plasma membrane, because by being able to insert into parts of the membrane, it makes them leaky, meaning that things leave the cell that shouldn't, things get into the cell that shouldn't. So having a good plasma membrane that is intact is key to success for the cell. So let's take a closer look at transport, how molecules are able to get in and out of a cell. So transport is the moving of molecules across the plasma membrane. And we can divide transport into two categories, passive transport and active transport. So we're going to talk about the features of passive transport first, and then the features of active transport. But really, they're just opposite of one another. So if you remember the features of passive transport, you're automatically going to know the features of active. So with passive transport, molecules move down a concentration gradient. Now, concentration gradient is a fancy way of saying there's a difference in the number of molecules outside the cell and inside the cell. So that's what a gradient is. It means you might have less of something over on one side and more of something on the other. You might have seen a color gradient where you have like your pastel yellow at one end and then it gradually gets darker or brighter, whatever the case may be, right? So it's pale on one end, but it gets much brighter on the other end. So it's a gradient because there's a difference in brightness. But with a concentration gradient, we're looking at quantity. We have a little bit over here, but we have a lot on the other side, okay? So molecules naturally want to go from where it's really crowded to where it's not so crowded. So they naturally go from where there's high amounts of the molecule to where there's low amounts. So that movement from going from high to low is known as going down a concentration gradient. And I bolded that for a reason. You're going to want to remember that moving down a concentration gradient means going from high amounts to low amounts. Because this happens naturally, no extra energy is required, okay? Things will just naturally go to where there's less of something available. So we're going to talk about three types of passive transport: simple diffusion, facilitated diffusion, and osmosis. But before we get into those details, let's just quickly mention the features of active transport. And again, it's just going to be the opposite of passive. So with active transport, molecules are going to move against the concentration gradient. And please cross out that word naturally. I apologize, that should not be there. Molecules naturally go down their concentration gradient. Okay, they do not naturally go against. So I apologize. Please cross out that second naturally right here where my mouse is. Okay, so with active transport, molecules are going to go against the concentration gradient. That means they're moving from low amounts to high amounts. Okay, so in this case, think of somebody like trying to fight through a crowd, right? They're going to have to exert a lot of energy to go against the grain to get through all the people. Okay, so in order to do this, an input of energy is going to be required. Okay, and for active transport, because they are going against what happens naturally, they will need the help of transport proteins all the time. Okay, so let's take a look at the different types of passive transport first. So we're going to talk about two types of diffusion. And with diffusion, these molecules do not need transport proteins. They are small enough to cross the plasma membrane. Well, let me kind of rephrase that. With diffusion, they don't always need a transport protein. So let's take a look at simple diffusion first. So molecules that can go across the plasma membrane using simple diffusion are normally small hydrophobic molecules, meaning they are compatible with that hydrophobic barrier. And so these molecules can literally just wiggle, wiggle, wiggle across that plasma membrane. They don't need any help, and no extra energy is required. So let's look at this figure and notice it's trying to show you this picture over time. So focus your attention where my mouse is right here, please. Notice all the little green hexagons are outside of the cell and there's none inside. So this means the concentration gradient, we have high amounts up top, low amounts down at the bottom. So with passive transport, remember, things want to go down the gradient from high amounts to low amounts. So they're naturally going to start crossing that plasma membrane. So now look in the middle. Do you see how two of them have already crossed over? And you can see a few here that are trying to wiggle their way across. And this process will continue to happen until there's about equal number of molecules on each side of the plasma membrane. Okay, so notice they crossed the membrane all by themselves. Again, these are normally small hydrophobic molecules. Another type of diffusion is called facilitated diffusion. So because you see that word diffusion, you know we're still talking about passive transport. Molecules are still going down the concentration gradient, but now they need help. That's what facilitate means, to help, right? My job as your professor is to facilitate your learning. I'm here to help you learn. I can't make you learn, right? I can't control what you do outside of the classroom, but I am here to help facilitate that process. This is where transport proteins come into play. These transport proteins are going to help these molecules move down their concentration gradient. So if you look at the top portion of this picture, notice there's a lot of the green molecule and there's 3 of the reds. Compare that to the bottom. There's only 1 red and a few of the green. So again, we have our gradient established. There's more up top, there's fewer down bottom. So as you can see by these arrows, the molecules naturally want to go from high to low. And so these molecules are going to keep crossing the membrane using that transport protein until the numbers even out. Once there's the same amount on both sides, then they'll be happy. Okay, so with facilitated diffusion, we tend to see this being small hydrophilic molecules or things that have like a charge associated with them, because these molecules are not compatible with the phospholipid hydrophobic barrier, which is why they need the help of a protein transport. I'm sorry, a transport protein. Getting all tongue-tied. All right. Our last type of passive transport is osmosis. So we're going to look at 3 different types of solutions because osmosis is the diffusion of water. Excuse me, I'm sorry, y'all. So we're going to look at what happens when you put a cell into 3 different types of solutions. Sorry, drink of water. All right, so let's look at a hypertonic solution. With a hypertonic solution, sorry, y'all. With a hypertonic solution, you're going to have more of a solute outside of the cell compared to inside of the cell. So you'll notice all those green triangles. Let's pretend that's salt. So we're going to place our cell inside of a salty solution. So there's more salt outside of the cell than inside of the cell. So what is going to happen? Well, it turns out water wants to go where there's more work to do, where there's more things to dissolve. So where is there more salt to be dissolved? Outside of the cell. So in this case, when you place a bacterial cell, we're talking about bacteria right now, water is going to rush out of the cell. Okay. When that happens, we see a phenomenon called plasmolysis. And so with bacteria, they do have a cell wall. We're going to talk about that in a future video. So the overall shape of the bacterial cell doesn't really change when water rushes out, but you do see the inside of the cell kind of becoming detached. The plasma membrane starts to detach from the cell wall. So it looks like the insides are shrinking, and that will kill bacteria, okay? So plasmolysis is the term we use for prokaryotes when they're placed in a hypertonic solution. When we talk about eukaryotes, though, because this same phenomenon will happen, osmosis, but in this case with eukaryotes, they do tend to shrink because like animal cells, for instance, we don't have a cell wall. So the cells would shrink. But when we're talking about prokaryotes, we use the term plasmolysis. Okay, so let's look at the other end of the spectrum, a hypotonic solution. So with a hypotonic solution, there's less of the solute, the salt in this example, in the watery solution than inside the cell. So we have more salt in the cell than outside. So again, water always wants to go where there's more things to dissolve. So in this case, water is going to rush into the cell. And when that happens, the cell is going to swell. But again, because of the cell wall that a lot of prokaryotes have, the cell won't necessarily burst, but it will get just a little bit larger, right? Because water's rushing into it. So for a prokaryote, we say the cell swells. Now for like an animal cell, a eukaryote, because remember I mentioned animal cells don't have a cell wall, the cell would burst, okay? And I'm just kind of tying in the eukaryotic stuff now because a lot of people have taken A&P, which tends to focus on humans, right? So animal cells. And so I just don't want you to be confused if you're like, no, cells burst in a hypotonic solution. Yes, animal cells do. Bacterial cells, it takes a lot to make them burst. Okay, so we normally just say in a hypotonic solution, a bacterial cell will swell. And then we have kind of the Goldilocks one, an isotonic solution, which is where there's the same amount of solute inside the cell compared to outside the cell. And so in this case, there is no rushing of water. Some water goes in while other water goes out. So there's really no change to the prokaryotic or eukaryotic cell. Same thing in this situation. All right, so now let's talk about active transport. We're only going to talk about this one really quickly. There's a lot kind of happening on this picture. But remember, active transport is the opposite of passive. So molecules are going to go against their gradient from low amounts to high amounts, and that is going to require an input of energy. So you'll notice in this picture, we have, let's see, 1, 2, 3, 4, 5, 6, 7, 8 green hexagons on the top compared to, what, 4 on the bottom. And so if you look at the first transport protein, you'll notice those green hexagons are trying to move outside of the cell. They're going from low amounts to high amounts. That requires an input of energy because they're going against the gradient. So you'll notice that ATP down there. Okay, that's the energy of the cell. So think about like a concert, okay? And if you came in late, right? So you're in the back where there's not a lot of people, but you're determined to get to the front of that stage where all the people are. You're going to have to channel your inner ludicrous and throw some elbows, right? You're going to have to exert some energy to go from where there's nobody to where there's everybody because you're fighting that crowd. That's essentially what's happening in active transport. And you'll notice if, again, we're looking at the top, which is outside of the cell, we've got 4 red ovals. We've got, 6 red ovals on the bottom. So again, with active transport, the potassium is going to go against the gradient. So it's going from where there's a little bit up top, and it's moving down to get to the bottom where there's more. Okay, and that also is going to require energy to happen. Okay, so let's wrap up this video with an apply what you know. As always, go ahead and push pause and do your best to answer this on your own. Then listen to me babble on about the answer. All right, you want to preserve your fruit salad by storing it in a sugary syrup. Is this a good method for preserving your fruit? Why or why not? Hint, think about osmosis. So I am a visual person, so I always recommend sketching out the scenario if you could. So first, it's going to draw a little bacterial cell. This is going to be a coccus cell, right? Because it's circular. Now let's draw the environment. I'm saying that we're going to put the bacteria into a sugary solution. So this is going to be a hypertonic environment because there's more sugar outside the cell than inside. So how you can remember that is if you're hyper, you have a lot more energy than normal, right? So in a hypertonic solution, the solution has more than inside the cell, okay? So remember, we're thinking about osmosis, which is the diffusion of water. Well, where's water going to go in this case? Remember, water always goes where there's more stuff to dissolve. So in this case, there's more sugar to dissolve outside of the cell. So water is going to rush out. That is going to cause plasmolysis. So remember, what that means is the plasma membrane, which is inside of the cell, is going to detach from the outer cell wall. And so that's going to cause the inside of the cell to kind of shrivel. And so the cell is not going to be able to work like it should. So this results in cell death. So that is why this is a good idea for preserving fruit. That's actually what they do in the grocery stores. If you go look at canned fruit salad, you'll notice some are stored in water, others are stored in a sugary syrup. And that's because by placing the fruit in the sugary syrup, if there was any bacteria still on the fruit when it got canned, it's not going to be able to survive in that sugary environment. So when we eat it, it's going to be dead and it won't do us any harm. All right, y'all, that's the end of this video. Please let me know if you have any questions.